Hello, good morning, good afternoon, and good evening, wherever you're joining from. My name is Jürgen Hummel, and I'm your host for today's webinar. I'm Senior Director Statistical Science at PPD, and I'm based in Edinburgh, in Scotland. It is a real pleasure to welcome you all today to our webinar, which has been jointly organized by PSI, statisticians in the pharmaceutical industry in the UK, EFSPI, which is the European Federation of Statisticians in the Pharmaceutical Industry, and ASA BIOP, which is the American Statistical Association, the Biopharmaceutical sections. And the topic of today's session is Estimands, and we have the pleasure of speakers from Europe and from the US representing regulatory agencies and the industry. The format for the webinar is that we will have 4 presentations that are followed by a panel discussion, which will be facilitated by my colleague Anna Berglind. If you have any questions, either on a particular presentation or for the panel discussion, then please put them into the Q&A chat, and we should have time for 1 or 2 questions on each of the presentations, but I will be mindful of the time in order to ensure that we have enough time for the panel discussion at the end. I now have the pleasure of introducing our first speaker, and that is John Scott. John Scott is Director of the Division of Biostatistics in FDA's Center of Biologics Evaluation and Research. Prior to joining the FDA in 2008, he worked in psychiatric clinical trials at the University of Pittsburgh Medical Center, and particularly relevant, he has been heavily involved in the ICH E9 Revision 1 expert working group in Estimands and sensitivity analysis. So welcome to you, John.
Thanks so much, Jürgen. It's really a pleasure to be here today to participate in this webinar. I'm looking forward to everybody's talk and in the panel discussion. What I'm going to be starting us off with today is a little bit of background about the Estimand framework, in particular, how it relates to implementation in the US regulatory area. And then I'm going to give some updates on the current status, both the sort of formal status of the framework and also some early experiences just from my own perspective. This is a disclaimer. The disclaimer is good, but what I would say is that this is a mixture of fact and opinion. Hopefully it'll be reasonably clear which parts are fact and which parts are opinion. So just starting with a bit of the prehistory of the Estimand framework in the missing data problem. So it's no mystery to anyone that missing data has long been known to be a problem in medical studies. For probably half a century or more, people have understood that the presence of missing data can lead to biased results and also decreased power and precision of estimates. Starting in 2008, the PDUFA 4, which was the 4th iteration of the law under which FDA sort of collects application fees, called on FDA to begin to address missing data in clinical trials in a formal way. At the time, I would say missing data discussions, that's also the year I happened to join FDA. At the time, missing data discussions were really focused on defining which analysis set people cared about, and often using fairly ad hoc methods for single imputation when missing data existed in the analysis set. It was also in many areas common to use last observation carried forward, primarily because this was thought to provide conservative test statistics in many cases. So what FDA did in response to this mandate was we commissioned a National Academy of Sciences study panel to investigate missing data in clinical trials. After several meetings and much deliberation, the NAS released a report or a monograph called The Prevention and Treatment of Missing Data in Clinical Trials. A lot of this was focused on the prevention part, on avoiding missing data whenever possible. And there was also a fairly robust discussion of analysis methods for missing data, including methods appropriate for various assumptions about the missingness mechanisms. But there was also a discussion of estimates, of what it was that the clinical trial was trying to estimate, and a discussion of the importance of sensitivity analyses in this area. In terms of estimands, what the panel observed, I think quite correctly, is that clinical trial practitioners were often pretty vague about what it was that they were actually trying to know. There would be a primary objective, and there would be sort of an understanding that everybody knew how to translate that primary objective into practice, how to implement it. And that led to people substituting sort of defaults like ITT or per protocol or other descriptions of analysis sets instead of spelling out exactly what it was they were trying to estimate. There were some translational issues going from the NAS recommendations to practice in drug development. One of the issues in terms of the methodologies, a lot of the missing data literature really originally came from survey statistics and designed experiments, and it wasn't always a good fit for the clinical trial setting. One reason for this is that it's, the recommendation that a good solution to missing data is not to have any. That's very intuitive, and in many cases is good advice, but in some cases it doesn't really make sense. For example, some missing data is formally meaningless. If somebody dies in a pain drug study, their pain scores after death are not missing, they're meaningless. They no longer are at risk of pain. There's also cases where data that's not missing has changed meaning due to an intervening event. So for example, and I think one of the talks later will go into detail on this example, if we're looking at overall survival in an oncology trial and patients cross over from one treatment arm to the other, perhaps after tumor progression, the meaning of survival in terms of the causal effect of the investigational drug has changed considerably. So this kind of consideration ultimately led to the formation of the expert working group for revision 1 to ICHE9 on Estimands and sensitivity analyses in clinical trials. And this document put a lot of emphasis on intercurrent events, the things that can happen after treatment assignment, which can affect the existence or interpretation of the variable you care about. And so underlying E9R1 was a framework, and the idea of the framework was it was meant to be a place to start off discussions for aligning planning, design, conduct, analysis, and interpretation of clinical trials. And what the document says is that the primary Estimand discussion should achieve alignment on 5 key attributes of an Estimand. Those attributes are what treatment condition it is that you're interested in, that you're studying. 2, what population of patients is going to be targeted by the clinical question of interest. 3, what variable or endpoint are you measuring for each subject to capture information relevant to that scientific question. 4, how intercurrent events will be handled. And finally, how the results will be summarized at the population level in terms of summary statistics. So again, as probably everyone on the webinar knows, there's a lot of focus on the handling of intercurrent events, and in particular on an enumeration of several different potential strategies for handling intercurrent events. Those were a treatment policy strategy, which many people understand to be roughly equivalent to the intent to treat principle. This is the idea that you use the value of the variable you collected regardless of whether the intercurrent event happened or not. One important thing about this, and which has led to some discussion in actual applications we've received, is that this does require that that value exists. You do have to have a meaningful value and you have to have collected it. Otherwise, you're in a different setting. Number 2, hypothetical strategy. So this is the idea of imagining an alternative world, a hypothetical world in which the intercurrent event not only did not occur, but could not occur, would not occur. The next one is the composite variable strategy, in which the intercurrent event itself is absorbed into the endpoint. This one is often used in cases where death interferes with the ability to measure another endpoint of interest, but that is also more widely applicable. The while on treatment strategy is less widely used. This is the idea of dealing with the intercurrent event by simply saying the value of the variable at the time the intercurrent event occurred is the endpoint we care about. And finally, the principal stratum strategy is a series of approaches for changing the population of interest to the population of subjects in whom the intercurrent event would not occur if they had been exposed to one or the other therapy, or both. So in terms of the current status of the document, the ICH guideline reached step 4, which is the final ICH draft, in November 2019. It's still undergoing the process of finalization throughout each of the ICH regulatory regions. According to the ICH website, I checked yesterday, it has been implemented in the EC. Andreas will be talking about that, in Canada and by Swissmedic in Switzerland. Not yet implemented in the US, in Brazil, and in the Asian regulators, China, Japan, Taiwan, Korea. For the US, what I'll say is that it's sort of in the final stages of publication, has been for a while. I think there's been quite a backlog of guidance publications for the FDA. I'm hopeful that it'll be published in the near future. In terms of what we're seeing in practice, there's been very substantial uptake of the recommendations in E9R1 among sponsors or applicants. We're seeing a lot of protocols come in with discussion of the primary Estiman. Sometimes it's very thorough discussion, sometimes it's more nominal discussion, but it's definitely in the ether. But there are still many protocols we receive that do not discuss Estimans at all. In terms of the ones that don't discuss Estimans, I would say that some FDA review teams currently have the practice of going back to the sponsor and the investigator and saying, please add a discussion of the primary Estiman so that we can talk about it. But that is not true in all cases. So we're sort of in this in-between stage of adoption. And I think there's going to be a lot more uptake to come over the next couple of years. One thing I thought to look at, I was aware of this guidance on the left, a guidance for acute myeloid leukemia. This is a draft guidance document, I should say. That had a discussion of Estimands for AML trials. And I did a quick look through FDA's recent guidances. Since ICH-E9R1 reached step 4 in November 2019, there have been about 20 clinical guidances that relate directly to the design of clinical trials. And of those, I only saw 2 that so far mention Estimands. It's these 2, the AML guidance and this esophagitis guidance. The esophagitis guidance in particular doesn't go into great detail. This isn't very surprising to me because the process of developing and publishing an FDA guidance is very long. So reacting to a step 4 document, you would have had to have started some time ago for it to have appeared in the last year. And also the E9R1 is not quite final in the US jurisdiction yet. I'm going to talk a little bit more about the AML guidance in another slide or 2. Having said that, there's a lot of uptake, and in that uptake, we've seen some growing pains. I would say one thing is that there are some elements that have led to communication difficulties between statistical and clinical teams. That's probably true both internally and externally. One area in particular, I think there's the nuances of what would be a hypothetical strategy versus what might be a principal stratum strategy are sometimes difficult to convey, and I will discuss that in a little more detail coming up. Another growing pain, it's not that uncommon for applications to come in where it's fairly clear that what's been done is a sponsor wants to do more or less what they've always done or what's always been done in an indication without really considering whether the Estiman makes sense, and instead sort of backfitting the Estiman framework to current practice. And I neither want to say that FDA review teams could do the same thing, but nor do I want to say that this is only a sponsor problem. It's at least theoretically possible that regulators could achieve the same goal. So just as a very theoretical example of what kind of thing I'm talking about, I'm sure everybody who works in clinical trials has seen protocols where the analysis set is described as modified ITT, which is taking an as randomized data set and removing some people for one reason or another. And I have seen applications come in where an MITT analysis set is being used, and it's being described as an as a modified treatment policy strategy. So that's the kind of thing I mean by backfit. Going back to that AML guidance and moving a bit into a discussion of hypothetical estimands, I copied this passage from the guidance because I think it's very interesting. What this passage is talking about is the idea that some people, it has been suggested, very passive voice, it has been suggested that a true treatment effect for a product for AML should be free from the influence of stem cell transplantation or other subsequent treatments. And so what they're discussing here is the idea of a hypothetical strategy for the introduction of HSCT or other subsequent treatments, and trying to address the question of what would the effect of the drug be if these treatments did not exist or were not available. And the guidance recommends not doing that. And it gives two reasons that I think are interesting. One is that you may not be able to design a clinical trial that allows you to actually estimate this well. If people in your trial are getting HSCT or other subsequent treatments, then estimating what would happen in a world where that doesn't occur is a difficult problem that raises causal inference issues. This wording is maybe a little more pejorative on the principle of causal inference than I would be, but it does make the correct point for sure that these are not trivial things to estimate. And then the second reason, the clinical relevance of such an estimand is still an open question if it can never be realized in practice. So if in practice people are going to move on to HSCT when clinically appropriate, it's not clear that imagining a world where that doesn't happen raises a clinically relevant question. So more broadly, some issues I've seen with the hypothetical strategy for intercurrent events. I think it's become a bit of a temptation in some cases to take a slightly naive approach to some fairly hard problems. A way of saying that would be, an intercurrent event is inconvenient, so let's say it just doesn't happen. That's very tempting. And I've seen it suggested that we could use hypothetical strategies for patients who drop out for adverse events. But what if that just didn't happen? Rescue medication, which is roughly what we're talking about in the AML case, or adherence are other cases where hypothetical strategies are sometimes proposed. For some of these questions, it seems like what people really want to know is more like a principal stratum strategy. We don't necessarily want to know what would happen if what the drug effect would be if AEs simply didn't exist, but we might want to know what happens in subjects who will not experience an AE, which is more of a principal stratum question. There seems to be a lot of reluctance to discuss principal stratum strategies, I think because people recognize this as a causal framework, and that scares people. I think it's scary. Having said that, it's not at all clear to me that doing hypothetical strategies well requires fewer causal assumptions than doing a principal stratum estimator. They both heavily depend on generally unverifiable assumptions. And one of the things I try to do is to think ahead to labeling and to communication. So imagine yourself telling a patient that the effect in the label is what they should care about. So just as an example, I accidentally repeated that bullet across 2 slides, but imagine that you're in a situation where rescue medication is very commonly used in clinical practice. You're introducing a potential new treatment to a patient, and you say, this effect, we know it has this mean effect among people who wouldn't need the rescue medication. So that's maybe interpretable. versus saying this is what the mean effect would be if rescue medication didn't actually exist in the world. I'm not sure that's useful information to a patient, and that makes me concerned about that strategy as an Estimands strategy in this case. Having said that, what I think the patient would like to know in most cases is their probability of needing rescue. Anyway, final slide. Having said all that, I took this picture from the Step 2 training materials on the ICH website for E9R1. And the basic idea of E9R1 was to create a formal framework for discussing Estimands internally and externally. I love this picture. One person says Estimand and the other person responds Estimand. I really like that. But I think we are seeing really good discussions, both internally and externally, as a result of moving the focus to this area. And it's been very good for everyone. So that is it. I believe I'm to turn the ball over to Andreas.
Thank you very much, John. Wonderful. Thank you for setting the scene and also bringing your perspective from the ICH E9 revision 1 so far from a US perspective. So we don't have any questions so far on that, so I think we'll move on straight to our second presentation, and it's my pleasure to introduce Andreas Brandt to you, a fellow countryman of mine. So he's been working as a statistical assessor in the Biostatistics and Special Pharmacokinetics unit at BfArM, which is the Federal Institute for Drugs and Medical Devices in Germany, since 2010. He's an expert of the Biostats Working Party at the European Medicines Agency, and before joining BfArM, he was a PhD student and postdoctoral scientist at the German Cancer Research Center in Heidelberg. So my pleasure, and over to you, Andreas.
Yeah, thank you very much for the kind introduction. So, sorry. Is everything fine now, or
Yep.
is it?
We can see it. Fine.
Okay, thank you. So thanks for the introduction. So and please note my disclaimer. And sorry, I have some. And as an introduction, so the ICH E9 addendum was published. The draft addendum was published in September 2007. Sorry, I have to say I have some problems to see my slides now. I'm very sorry. Okay. So let's start. So the draft ICH E9 addendum was published in September 2017, and now in the meantime, the final version of the addendum was adopted by CHMP in January this year, and it came into effect in Europe in July this year. And so since the publication of the addendum, we had the possibility to gain first regulatory experience in all three major areas of regulatory activities. So that is during the assessment of marketing authorization applications, during scientific advice, and we also started to include the framework in regulatory guidelines. So, and before I want to go into the details of that, I want to start with some general observations. So what we can definitely say that the estimate framework has arrived in the system. So nowadays we receive almost no new plans for confirmatory studies that are planned without using the framework estimate, or at least the term estimate is mentioned somewhere in the protocol. But what we also see that there's a lot of variation in experience and understanding with regarding the framework. So first of all, there's a lot of variation between companies. So there are some companies who adapted the framework early and have a lot of experience and challenge regulators with sophisticated proposals. But on the other hand, as John also already mentioned, there are the companies who just translate what they're always doing into the estimate framework because they feel it's a regulatory must. But of course, there's also still some variation in experience between regulators, so in Europe between different regulatory agencies, but also within agencies, there's difference in experience, for example, in particular with regard to clinical assessors. And what we also see is that there's a lot of variation between therapeutic areas. So in some therapeutic areas, the framework is adapted, so mainly in those that were the initial motivation to start thinking about estimates, such as diabetes or neurology. But in other areas, such as oncology, there's more reluctance to adapt the concept. So another interesting observation that I made during discussions with the industry is that the perception of the addendum is partly quite different between industry and regulators. So the industry mainly sees new possibilities. So for example, that the addendum allows deviation from the traditional ITT analysis. And so there's a lot of interest in hypothetical strategies or principal stratum strategies. But on the other hand, for me as a regulator at the beginning, it was more going back to the roots, so that we say that we insist on a treatment policy strategy where we implicitly accepted sometimes hypotheticals in the past, so by accepting statistical methods that address a hypothetical question. But of course, as always in life, the truth is probably somewhere in the middle. I think we have to acknowledge, yes, the estimate framework offers also new possibilities and thinking. But as always, any change from current standards needs to be well justified to be the best solution for design and analysis in the current situation, and not just because it's something new that we want to try. Now, going in some of the more details of our experience. So interestingly, many of our early experiences with Estimands do not come from study planning or scientific advice, but from the assessment of a marketing authorization applications. Of course, at the moment, we are in a transition phase. So many clinical studies to support marketing authorization applications that we assess nowadays were planned without using the estimate framework because they started before the addendum was published. But of course, we can still ask what estimate was actually implicitly targeted by the primary analysis. And so we see frequently that companies take that up and try to define this estimate retrospectively, but also regulators think about what estimate was actually targeted and what could be alternatives to the targeted estimate. But of course, there are some limitations of this approach when the study design was not really aligned to the estimate that we would consider of primary interest. So then the issues that are raised are of course, primarily, is the implicitly targeted estimate really the one of primary regulatory interest? Then you look what intercurrent events actually occurred and what was their frequency, and sometimes you have to ask questions about that because it's not that clear. And then we have to look where the strategies for handling intercurrent events are appropriate from a regulatory point of view. And this leads frequently to asking for additional sensitivity analysis and often also to supplementary analysis targeting at a different estimate than the one that was used as primary estimate. And this finally leads to the question, what is the appropriate estimate to be included in section 5.1 of the summary of product characteristics, where the results of studies are described in euro. And this this is often not the primary estimate as it was included in the protocol, but we often insist to include another results for another estimate that we feel that is of greater relevance. Of course, questions around estimates are also a frequent topic during scientific advice procedures. So usually if companies do not raise it when they ask for design of a phase 3 study, usually regulators ask them to take that up. And here we really have to say that we see that the estimate framework serves its purpose. So it really provides a common language to phrase the question of interest, primary interest, and it serves and we see more transparency. That's a question of clinical interest is really clearly defined, and it's not implicitly defined by some choices that are made for statistical analysis that are hidden somewhere in the statistical analysis plan. But having said this, we still see, of course, we still see some deficiencies when the framework is applied. And we also see disagreements regarding the estimate that should be targeted. And there are also often issues regarding the alignment of the estimate with the design and the estimator. So regarding to deficiencies that we frequently see, so this is firstly that the estimate is specified fairly late at a fairly late stage of study planning. So we don't see an estimate, and when we ask the company, they say, oh, that's something that we will define in the future. But of course, this is not optimal because the estimate definition is crucial for choices that need to be made for study planning. Then often the estimate is considered as a purely statistical issue. So for example, in the protocol, it's just stated, yes, the estimate will be specified in the statistical analysis plan. Then we also still see mixing the definition of the estimate and analysis issues. So this concerns mainly questions around missing data handling. So missing data is still often considered as an intercurrent event, which it is not, or the missing data handling is even considered as a part of the summary measure to describe treatment effect. Then we also see incomplete lists of intercurrent events. So the companies focus only on just one event that they are particularly interested in and forget that there are more that need to be addressed. Or intercurrent events lack granularity, so it's just that use of rescue medication or discontinuation of treatment when the reasons for taking rescue or the duration of taking rescue or the reasons for discontinuation may be of importance. Then often sometimes we see so one strategy fits all solutions, so that the same strategy is used for all intercurrent events without a real proper justification. And sample size calculation is often also not aligned to the estimate, so it is not considered that the treatment effect may be smaller when a treatment policy strategy is targeted. So disagreements often occur regards the question, what is the estimate of interest? So companies tend to propose hypothetical or principal stratum strategies, somehow often for the wrong reasons. So in the meantime, I have quite some routine in writing that the pharmacological effect or the effect if patients follows the instructions on the label is not the effect of primary regulatory interest to make a benefit-risk decision. So an interesting question is regarding the adequacy of the treatment policy strategy for non-inferiority studies that is sometimes proposed. So this is an ongoing discussion, but of course there are some issues regarding sensitivity in this case. Then we have also issues with regard to the alignment of the design and analysis with the estimate. So what is really a good thing that we see nowadays changes in study design. So in particular, at least in the protocol, it is a better follow-up is specified, so patients are planned to be followed irrespective of treatment discontinuation. And we also see changes with regard to the statistical methods that are routinely used that aim to better align the estimate with and the estimator. So for example, where a few years ago mixed models for repeated measures were frequently used, we see nowadays that reference-based multiple imputation is developing towards a new standard missing data approach. Of course, this applies particularly for treatment policy strategies. And we also see that the traditional censoring rules are changed to be better aligned to the estimate. But sometimes, of course, we also see some disalignments of design and analysis with the estimate. So this applies, or we have disalignment of design and estimator. For example, where we frequently see that missing at random strategies are proposed or to address missing data, although a treatment policy strategy is pursued. So the company still plans to follow patients after discontinuation, but data that is nevertheless missing is still replaced under a missing at random assumption, which is of course not appropriate because the potential loss of effect after that is not taken into account. But of course, in some cases, we have to acknowledge that there's something like what I would call the estimator gap. So in some cases, there are no well-established methods for the estimation of the estimate of interest in some areas. For example, for time-to-event analysis, when we want to use a treatment policy strategy, and we have missing data, censoring after study discontinuation may not be adequate, but we do not really have good alternatives to that in the moment. So what is and as I mentioned in the beginning, we also started in Europe to implement the estimate framework in therapeutic area specific guidelines. So for guidelines that include the estimate framework and that are already finalized are those for Alzheimer's disease, Crohn's disease, and ulcerative colitis. While other guidelines documents that include the estimate framework are not yet finalized and are or were in the public consultation phase, for example, those for diabetes mellitus or chronic non-infectious liver diseases. So this work was unfortunately interrupted and will be continued. So it was interrupted due to the EMA relocation from London to Amsterdam, and of course, no, because the pandemic changed some priorities. So in the future, we will also update some statistical guidelines to better align to the ICH and 9 addendum. And there are also some new areas such as real-world evidence and registries where the estimate framework may also prove to be useful. So now, what are our learnings from the work on therapeutic area specific guidelines? So first of all, what we always try to do is to separate the estimate section and the statistical analysis section to clearly highlight that these are two different issues. And the estimate section is really a joint work of clinicians and statisticians. So we try to identify together the relevant treatments to be compared and what could be the intercurrent events in this specific therapeutic area. And then we explain the different strategies to the clinicians, but of course, finally, it's up to the clinicians to decide on what questions they are interested in. So how the specific intercurrent events should be taken into account. And once they understood the concept, clinicians usually have clear ideas what strategies they prefer. So for example, for treatment discontinuation, we usually see that they prefer treatment policy strategies, while for rescue medications, hypotheticals are often preferred, but not if rescue was not taken, but if patient had not taken rescue but would have discontinued treatment, or they want to use composite strategies. But when discussing with clinicians, we also face some challenges. So we still see some reluctance to adapt the estimate concept in some therapeutic areas. And I think partly this is also explained by different traditions to handle intercurrent events and missingness in different therapeutic areas. So by those areas that usually use continuous endpoints, missing data was always considered as some kind of nuisance, and it was usually left to the statisticians to care about that. While for other areas where time to event and responder endpoints are used, there are often already strategies in place to handle missing data and intercurrent events. Although, of course, that was not this terminology was not used yet, and it's not completely the same thing. But still, there's some reluctance from clinicians to change these well-established approaches. So clinicians really need to be convinced from the added value that the estimate framework provides, and this works best when you really have specific examples to explain it. So now coming to a conclusion is, so to my experience, the addendum really fulfills its purpose by providing a framework for a common language to phrase a question of interest in a transparent way. It leads often to a better alignment of study objectives with design and analysis, but still we need more experience in many areas. Yeah, and so we still have to overcome some specific problems. So for example, the estimator gap sometimes requires new thinking and maybe also the application of new statistical methods. We need more collaborations with clinical clinicians to really that the concept is generally adapted. And so I would say it's a promising beginning, but it's still a long journey until the framework will be fully adapted. So thank you very much for your attention.
Perfect. Thank you very much for that, Andreas. We've got a question here to maybe pass on to you specifically for that presentation. And the question is, you said considering Estimands, hang on a second here. You said considering Estimands as a purely statistical issue, we have noticed that the most of the questions we get about Estimands come from the statistical reviewer. Is the Estimand being considered more than a statistical issue by the agencies? What are you doing to make sure the clinical or medical parts of the agency are using the concept?
Yeah, that's really a good question. I think it's true, though. Now, still more. I think most thinking about estimate still comes from the statistical assessors, but I think we, at least from, I can speak for myself, I think we at least try to involve the clinicians. So we explain them the problems and ask them what they think about that, or what they prefer, and we try to take them on board. But it's true that usually they don't come with estimate questions from their own initiative yet. So we really have to engage them.
So I guess still work in progress.
Yes, definitely.
Okay, thank you. We've got a second question, but I think we'll keep that to the panel discussion. So thank you very much for that, Andreas. And we'll now move over to the next speakers. So it's my pleasure to welcome Evgeny Dektyarev, who is a global programming biostatistics head leading a team of quantitative scientists in hematology at Novartis. He joined Novartis in 2013, and most importantly, he has co-founded and co-leads the industry working group Estimands in Oncology in 2018, which has since become an EFSPI PSI special interest group and an ASA Biopharmaceutical section scientific working group. So, Evgeny, over to you.
1 second. I have difficulties to see the slides now. This field. Okay, maybe I can do it. Can you see my slides now?
Yes.
Okay. So hi everyone, I will share today some thoughts on the opportunities created by the ICH9 addendum for oncology drug development, and throughout my talk I will refer to some activities of the industry working group that Jürgen just mentioned, and so I would like just quickly to introduce it. So we've co-founded it together with Kaspar Rufebach from Roche more than 2.5 years ago, and at that time the purpose was to discuss the impact of the draft ICH9 addendum on oncology. And from the beginning there was a lot of interest from the industry, with now almost 30 companies being involved, and also from the regulators around the world. And later on, as already mentioned by Jürgen, we received the status of both EFSPI PSI Special Interest Group and ASA Biopharm Scientific Working Group, and we are happy to contribute to this joint webinar today. You can find more information on our homepage, but I would like just to highlight 2 recent events, the webinars that we organized in collaboration with EFSPI as well as Basel Biometric Society. One in June with clinical speakers on estimates in oncology. I think maybe, at least to my knowledge, the first webinar with clinical speakers, and the second one in September on causal inference and principal stratum, also with academia speakers. And if you didn't have a chance to watch them, then the recordings are available, and I would really encourage you to take a look. And I will refer later also to the principal stratum webinar there, but maybe just one word on the clinical engagement, because it remains high on our agenda. And while we've had some small successes with this webinar, but also with an ESMO poster presented by a key opinion leader, and also a recent acceptance of a manuscript on Estimands and adjuvant RCC, again co-authored with clinicians. There's certainly a lot to be done. I fully agree with Andreas there, and I would be interested also how the industry and the regulators can also work together to engage the clinicians on both sides as well. So maybe if there are any things to share by Andreas and John, this would be very interesting, I think, for us. And one of the reasons why I've co-founded the working group was actually my belief that the estimate framework can help us to improve the current public perception of oncology drug development, of regulators and sponsors in particular. And the current situation is the following, I think, from my perspective. We have seen some advances in recent years with new therapeutic modalities and exciting therapies like immunotherapy, cell and gene therapies, radioligand therapies that produce durable responses in many indications. And we do see more and more ongoing clinical trials, which I think is great for patients. But on the other hand, regulators, sponsors, and payers are actually criticized for granting approvals to drugs that seem not to improve survival and that are very expensive. And on this slide, you see just 3 very recent publications. You see the dates: October, September, November. So it's all really the last 3 months, suggesting that there is ambiguous evidence of survival benefit in oncology trials, that progression-free survival, the main endpoint in many of our trials, is actually a rather bad surrogate for overall survival. And on the left side, you also see a conclusion of one of the publications suggesting that early and less complete evidence on benefit risk was sufficient to grant regular approval. And this is raising questions actually over regulatory standards for the approval of new medicines. And they reviewed their EMA and FDA approvals in the last years. And I think we are all also aware of the BMJ publication last year, where that also led to many follow-up reports in the media suggesting that our oncology trials are poorly designed or even flawed, and that half of new cancer drugs actually doesn't show benefit for survival or quality of life. And I think one of the reasons for these publications is actually the non-significant result for overall survival, because people look at overall survival, sometimes even not checking whether the study was powered for overall survival. And if there's no benefit observed, no significant benefit, statistically significant benefit, then they conclude that overall the drug does not improve survival. And certainly then it's also difficult to establish correlation maybe between PFS and overall survival. I refer that to the previous publication on the slide there. And what we also see quite often there, that actually most of the time the main reported result for overall survival is the treatment policy estimate. And in particular, we have an issue there with non-significant results when subsequent therapies actually do not represent clinical practice. And I will just quickly illustrate what I mean there on the next slide, in particular for those of you who may not be familiar with oncology setting. And so on this slide, you see a traditional design, drug A randomized versus standard of care. And typically patients after discontinuation of study treatment would receive other antineoplastic therapies and would be followed up till death. And usually we analyze overall survival using treatment policy strategy. Using treatment policy strategy, which also means that we capture the effect. I'm not sure. That we capture the effect on the choice and impact of subsequent therapies. And I just want to note here that maybe in the past, we would have actually expected to see balance of subsequent therapies, But I think nowadays we don't really expect to see it in most of the indications because there is much more choice now, and actually many therapies are being investigated specifically in patients who maybe progressed on a certain therapy, and therefore physicians really need take decisions on subsequent therapies in light of previously administered therapies. And so even if your study is maybe blinded, quite typically, you would unblind after progression so that physician really can take the best decision for the patient with regard to subsequent therapy. But even if there is maybe no balance in subsequent therapies, as long as the subsequent therapies represent clinical practice, the treatment policy estimate will be clinically meaningful. On the next slide, you see another typical scenario where drug A is maybe already approved as next line therapy after standard of care. And now the sponsor is moving drug A into investigation in early line. And so patients who progress on standard of care can actually receive drug A on the market. And in this case, again, this crossover actually reflects clinical practice, and therefore treatment policy overall survival estimate remains interpretable at the time of the readout. But what about a situation when patients from standard of care after discontinuation actually receive drug A or maybe drugs with the same mechanism of action, which is not yet approved as next line therapy after standard of care? And this can happen in a study where crossover is allowed upon progression per design, or maybe because after successful primary analysis with PFS, patients start to switch and maybe protocol amendment is implemented to allow patients actually to switch to drug A. And in that case, the choice of subsequent therapies would actually not reflect clinical practice. The treatment policy estimate in this situation would actually compare drug A followed by standard of care or other available therapies versus standard of care followed by drug A or other available therapies. And this doesn't seem to be very relevant for a patient or physician if we compare it against the standard of care followed by drug A, because drug A is not approved for the subsequent line after standard of care. And so patients don't have access to it. And in such specific situation, I think the benefit on survival without crossover possibility may be more informative to physicians and patients. And this would be the hypothetical estimate, and I will share some thoughts on that in a minute. But maybe just to summarize a few points where I think I see the most benefit of the framework, and it is really about, I think, improving communication. Communication between regulators and sponsors to discuss alternatives for main overall survival analysis, addressing relevant questions for patients and prescribers. And certainly, I think treatment policy estimate will be the main question in majority of the settings and majority of our studies. But there will be situations where the hypothetical estimate is likely to be the more relevant one. And in that case, I would hope that we will try to address this question for patients and prescribers. There's also, I think, an opportunity to communicate the value of our drugs better in publications, but I think there's also an opportunity to improve communication between physicians and patients by improving the overall survival description in our labels. And I have here just two examples, one from US and one from Europe, actually quite an old one in US, and this is really just demonstrating that this is not a new issue, and estimate framework is not really adding complexity, but it rather provides us a tool to discuss already existing complexities. And so what is interesting about these two labels is that in both of them, the description of overall survival is quite similar. First, there's a statement on the hazard ratio, or there's even a statement that there was no statistical significant difference between two arms. And then what follows is a description of subsequent therapies that suggests that there's a very high number actually of patients who are crossing over that could potentially confound overall survival. And I think we talk a lot about patient-focused drug development. And I think patient-focused drug development also means that we do address questions that are relevant for patients, and we ensure that it is also appropriately described in the label, so that physicians can actually inform the patients about the answer to this question. And certainly overall, the benefit on overall survival is one of the key questions for patients. And I think here we really have a lot of room to improvement to do something more meaningful for patients. Oops, sorry. Sorry. Yeah. So, and the hypothetical estimate can represent one solution to that in these specific situations. And I think just to remind everyone, I think, and John already spoke about this, that yeah, ICH9 addendum acknowledged that there are some hypothetical scenarios that are likely to be of more clinical or regulatory interest than others, and that hypothetical estimates have been actually often targeted as primary analysis in pivotal trials. In oncology in particular, because it was implied by the FDA guideline that suggested censoring for new anti-cancer therapies. And Andreas also mentioned the Alzheimer and diabetes guidelines, where the EMA suggests the use of the hypothetical estimate. And in this situation, I think the overall survival benefit without the crossover possibility certainly represents a key question for patients and prescribers. And we do have methods to address it if properly planned. And yet, I think this hypothetical estimate is actually facing some headwinds at the moment, as these methods rely on assumptions, as all other methods do. And I think John referred to this in his presentation, saying that these are not trivial things, and I fully agree with that. But these non-trivial things are addressing maybe the much more relevant question to patients and prescribers at what we are currently doing. And so I think if we acknowledge that, then it really provides us a great opportunity to learn together and to collaborate also with academia to address important questions for patients. Another area of communication where certainly the estimate framework has a lot of potential is to improve health authority interactions and to ensure that we get the questions right and that we understand each other better. And for that, I would like to refer to a case study that was actually presented in the recent webinar on causal inference that I mentioned before. And my colleague presented their CART study where commenting on the protocol, FDA actually requested us to censor certain observations in a supplementary analysis. Now, what we realized was that this censoring would actually likely target a hypothetical scenario that is not very meaningful for clinicians or regulators. And we felt that FDA is probably actually more interested in the principal stratum estimate. And so we proposed the principal stratum estimate to them. And indeed, FDA agreed to use the principal stratum strategy there as supplementary analysis instead of censoring. Was it more work for us to propose the principal stratum, to describe the analysis and the assumptions? Absolutely. It would have been much easier to censor the observations. And maybe this is actually what we would have done in the past without the ICH9 addendum. But I guarantee you that it's also a much better feeling if you know that the analysis will address in the end the clinically meaningful question, compared to the situation when you know from the beginning that this censoring will not be relevant, actually provide a relevant answer to patients, or this treatment policy estimate for overall survival in a situation with crossover will actually not be meaningful and interpretable for physicians. And principal stratum is certainly another opportunity to learn together and to collaborate with academia, because we do see questions around principal stratum everywhere in drug development. And many examples were actually recently summarized in the paper by Björn Bornkamp and the causal subteam of the industry working group that you see here. And you will hear about another application of principal stratum in the talk by Vlad that is coming next as well. And just briefly to mention two other opportunities. There are many more, but I think certainly from the industry perspective, I think harmonization of therapeutic guidances would be welcomed, and in particular in oncology, we have different approaches between EMA and FDA with regard to censoring of new cancer therapies, and maybe the framework can help at least to have a discussion about this and to understand also better the different approaches the agencies are taking there. And hopefully also we will see more focus in the future in the guidances on questions of interest and less on the censoring rules, that I think is yeah, more the case at the moment, at least for oncology. Another area where maybe that not so much discussion happened so far for an application of Estimands framework is the real world evidence. And I think there the framework really could provide us a tool to support structured discussion about the question of interest and the planned comparison between single-arm trials and real-world data, or if you supplement your control in a randomized trial, and really have a transparent description of population endpoint, handling of intercurrent events in the two sources. And if we really do it using the framework and describe the potential limitations and also uncertainties, then I think it would also help the regulators to take the decision whether the real-world data source is suitable for comparison, and it would also certainly allow the sponsor maybe already at the beginning to realize whether this source is suitable or not as well. And actually, Estimands framework is very similar to the target trial framework that was proposed by Miguel Annan for the observational studies there. And this topic, real-world evidence and estimates, is actually one of few topics that we have just started to discuss within the industry working group. And you see here some other new task forces that we are kicking off. And if you're interested to join, it's certainly still possible and everyone is welcome. And as you see, it's a broad range of topics, and we even start to think about things that we used to do, I think, in a very traditional way, just copying from one study to another, like median follow-up. And this has been discussed in the follow-up quantification task force. But what are we actually trying to answer with the median follow-up number, and how do physicians interpret this? And I'm not sure whether everyone has actually the same understanding what this number means, and maybe it's useful just to go back to the roots and to have a discussion there with the Estimands framework as well. And so to conclude, I think the framework really provides us an opportunity to improve also the perception about the approved drugs, and also to do more patient-focused drug development, really ensuring clarity and interpretation of results, and also describing the added value of our drugs better in the publications and in labels. And hopefully we'll see also more dialogue about that in future. And I think it's also really a big opportunity for sponsors and regulators to learn together and to develop best practices, for example, for the approaches for the to tackle hypothetical estimate for overall survival, and really to share experience and to collaborate with academia to ensure that we are able to address important questions for patients. And we've done it successfully before, for example, in oncology with Bayesian designs for dose finding, and instead of traditional 3+3. And I think we need another maybe paradigm shift there as well, moving from one size fits all approach with ITT to really doing maybe better science on a case-by-case study. And I would like just to acknowledge many colleagues in the industry working group, as well as at Novartis, with whom I had discussions over the last years. And on the last slide, you just see the links to the recordings of the webinars that I mentioned, and the publications of the working group that cover a broad range of topics from COVID-19 and principal stratum to more case study discussions. And I invite you to take a look once the slides are uploaded after the webinar. Thank you.
Excellent, great. Thank you very much for that, Evgeny. Very interesting, and I like your sort of conclusion and outlook about collaborating there together. We've got one question that came in specifically for your presentation, and it says, is it possible that there could be estimands on which we may want to base hypothesis testing for regulatory decision-making, and others which may be more useful with regards to communication of results to patients and prescribers?
I think, well, certainly the non-proportional hazard setting is probably one where I think people are discussing such things, and we do realize that you need more than just one number to describe the treatment effect, right? And the hazard ratio may not be the only metric that you would provide to physician and patients, right? And that you really need other aspects as well. And so certainly there, I think we already understand that there's a need to have several estimates in a way to describe different treatment effects. And I think actually Andreas mentioned in the previous presentation as well that there are some discussions within the CHMP and EMA on which estimates should be covered in the label, and maybe we can also touch base on that in the discussion. But I would be interested to hear also regulatory perspective on that.
Perfect, great. Thank you very much for that, Evgeny. And with that, I am now have the pleasure to introduce our fourth speaker. So welcome to Vlad Dragulin. He's Vice President, Scientific Fellow in Quantitative Sciences at Janssen's pharmaceutical companies of Johnson & Johnson. He joined Janssen in 2014, and he is also a pharma representative on the ICH E9R1 expert group, and he's also a member of the Pharma Clinical Development Work Group. So Vlad, welcome, and over to you.
Thank you, Jürgen. And I want to thank the organizers of this webinar for giving me the opportunity to present. So I hope that you see my slides. This is the, yes. I plan to talk about special considerations of Estimands framework in COVID-19 vaccine trials. For transparency, all leading pharmaceutical companies that are running now COVID-19 vaccine trials made their protocols public, available. So I use this opportunity just to look how different companies are focusing or emphasizing the Estimands in their protocols. So that is the scope of my presentation. So I will just go over these 4 protocols and share with you how different companies are defining Estimands for vaccine efficacy. Then I will spend some time, that will be the core of my presentation, about causal estimates for vaccine efficacy in prevention of disease and causal estimates for vaccine efficacy against severe disease or death. So here are the treatments that have been already investigated, or the trials are ongoing, everybody knows. So you can see here that primary efficacy objective in all these 4 protocols is to evaluate, or sometimes they use the word demonstrate or estimate, the efficacy of investigational vaccine in prevention of COVID-19. And AstraZeneca has a treatment which is 2 vaccinations or 2 injections of their drug versus placebo. Janssen is another type of treatment, but is a single dose of their vaccine versus placebo. Moderna also has 2 doses versus placebo, and Pfizer also 2 doses. So it's not that they will investigate 2 doses, it's 2 injections of the vaccine. Sometimes it's 28 days apart, sometimes 21 days apart. So roughly speaking, it's a 2-arm type of study, but there are different sort of regimens for vaccine from one company to another. So population, this is the second then component of the estimate. AstraZeneca formulates this as participants who receive at least 1 dose of study intervention, and in the patients who are seropositive at baseline, analyzed according to their randomized treatment. Janssen is reducing a bit the population definition, is saying participants who receive study vaccine and who are seronegative at the time of vaccination and who have no prior major protocol deviations that were judged to possibly impact the efficacy of the vaccine. Similarly is Moderna, participants who received planned doses of investigational product per schedule and have no major protocol deviations. And Pfizer is saying participants without evidence of infection before vaccination, which is kind of similar to seronegative at randomization, and included in the available efficacy population and in the all available efficacy population. So let's focus on the variable. So here there is a difference between how different companies in their protocol are defining the variable. So AstraZeneca is saying this will be the first case of SARS-CoV-2 RT-PCR positive symptomatic illness detected. Sorry, I have to take this out. I have to do it because it's sitting on my guys. Okay, so illness occurring 15 days post second dose of study intervention. So remember, their strategy was to do 2 injections. So they defined the variable as first case of this infection, if you like, and this is how they will define it is infected or not, this RT-PCR, but occurring 15 days post second dose of study intervention. This is very important. Janssen is saying first occurrence of molecularly confirmed moderate to severe critical COVID-19. So in AstraZeneca, they don't say what kind of level of infection they will see. So Janssen is a bit more specific. They are looking for moderate and severe or critical. Moderna is first case and is at least 14 days post-vaccination. Remember, in time, on the patient level, this is just 14 days after the vaccination. This one is after 6 weeks after vaccination, because you have 2 vaccinations 1 month apart and then another 2 weeks. So Moderna is another definition of symptomatic illness occurring 14 days after the second dose of study intervention. And Pfizer is slightly different. Yes, they also do have 2 vaccinations, but they will count the variable after 7 days only after the second dose. So nota bene here, I want to emphasize right away that then in the protocol later on, roughly speaking, they are proposing to use protocol analysis in a sense that subjects that have their event, one of these events, before that time tau, for different companies, they have different definitions of this tau, so most of them 14 days after the second vaccination or the first vaccination, those patients will be excluded from analysis. So the focus is what is happening to the subjects with the infection after, let's say, 2 weeks after the end of the treatment, if you like. So I will come back to this a bit later. And then summary measure, of course, in vaccine efficacy, there are 3 possible incidence measures of infection. So one is called cumulative incidence, or in the vaccine terminology is called attack rate. This is a risk of seronegative subjects will get infected during fixed follow-up period, let's say 2 months. So as here in this definition is survival function. So another one is incidence rate, or risk of experiencing an infection during a given time unit, let's say per month, or per week, or per year. And the third one is hazard rate, as in many time to event type of situations. So that is a definition of hazard rate. And then using this potential 3 different incidence measures, they come up with 3 definitions of what is summary measure or vaccine efficacy. So vaccine efficacy for the first one is 1 minus RR. Of course, relies on a very crucial assumption that each subject will have fixed period that they will be observed. Second one is assume constant incidence rate, let's say constant incidence rate per month, depending on the time, and is defined as 1 minus ratio of this incidence rates. And the third one you can say is 1 minus hazard ratio, which is more useful, more frequent in other therapeutic areas where time to event is the primary endpoint. And of course, this one we know that it is assuming constant hazard ratio, because you can see here on the left, there is no T, it's assuming that this hazard ratio will remain constant during the period of observation. And then intercurrent events. I put here a list that I picked up from the protocols, but my presentation will be focused on number one, and let me explain how I want to investigate that one. So, like I said, in the definition of the variable, the protocols were saying that the variable will be counted, or event will be counted only after tau days after the second vaccination. For tau is either 14 or 7, or for Janssen, because it's just 14 days after the first injection. So there are other, of course, intercurrent events that they are indicating in their protocol. Let's say missing dose of investigational product. This is specifically important for 2 injections regimens. So it may be that subject was randomized, they got first vaccination, but they didn't come to the second vaccination. And that is then an intercurrent event that we need to deal with. Another one is seropositive at baseline. When the subjects are coming in the study, the tests that we have today are not very timely. So it may be that they will be randomized either to vaccine or placebo, but later when those results of the test will come, it will be determined that they were seropositive at baseline. So we are not interested in evaluation of vaccine efficacy in seropositive, so we need to deal with this type of intercurrent event. Luckily, this is hopefully balanced by randomization, and it doesn't cause a big problem in the evaluation, but it is just highlighted there. So the other one is death, and we know that death is always very complicated. And in this case, the subject may be dying due to COVID, but because we are enrolling a lot of subjects with comorbidities, they may be dying during the follow-up due to other than COVID-19 reason. So then how do we deal with that intercurrent event? And then withdrawal from the study is enumerated in several protocols, but we know, according to ICH-E9, that this is a missing problem data, not an intercurrent event. I just put it for complexity. Okay, so now let's focus on strategies for intercurrent event number one. So, of course, they are using conventional terminology. So per protocol analysis, this means that the subjects which have event during the first tau days will be excluded from analysis. So, of course, we cannot rely anymore on balance by randomization in the comparison between vaccine regimen and placebo regimen. And we need to be careful how we will provide an estimator for the estimand, because that estimand, like Evgen was mentioning and before, it might not be an estimand of interest. So the resulting estimator is biased for the causal estimand, and I will show later 2 potential definitions of causal estimand in this situation. An alternative one is ITT analysis. Sometimes they use MITT, like John was explaining. That means is vaccine efficacy analysis will include all randomized participants, irrespective of the time of experiencing the endpoint. So if a subject had an event before time tau, they will be counted with their time to event. So some of the companies are picking one as a primary, another one as secondary or supplementary. And of course, there is the third one, which is related to that C word that John mentioned, is principal stratum. So that means that we want to deal with this intercurrent event, but we define that we are interested in evaluating vaccine efficacy that will include all randomized participants and will evaluate vaccine efficacy in the subset of population who would not experience the endpoint within the first tau days on both treatment arms. So probably all in the audience are already familiar with the principal stratum, and you know that this is different from per protocol. Because in per protocol, you just know what is happening in one of the treatment arms, but not in the both. So let's give you a bit more details. I don't want to bother you too much with the formulas, but because you have the slides available after the presentation, I have to quickly go through these definitions. Let's T, capital T, be random time from randomization until the COVID disease event. Then we may have censoring time, as usual, censoring in time to events. So we will observe Y, which is minimum between T and C, and this indicator that indeed it was an event, but not a censoring. And let A be an indicator of adherence or compliance to assigned treatment. You remember there, for 2 injections regimens, it may be that some subjects will be not adherent or compliant to the 2. So let's say within the first tau days, we treat A as undefined if T is smaller than tau. Let Z be the treatment assignment, V or P, vaccine or placebo. And then for each subject, roughly speaking, we can imagine that we have 2 potential outcomes of this vector of observations. And they are denoted by Z index there, or Z can be either a vaccine or placebo. Now, the PS cohort of interest, the principal stratum we can define might be this way. So this is indicator of all participants which have time to event greater than tau, and they were compliant. So this is subjects who would not experience the endpoint within the first tau days on both treatments and who would adhere compliant to both treatments. So then what is the causal vaccine efficacy effect, or the summary? You can define this survival function, and you can see here that is conditioned that these events are after tau, where tau is the time. You can define that then hazard ratio for this type of definition of survival, and you do this for each of the potential treatments for vaccine and placebo, and then you define causal vaccine efficacy effect using, let's say, the third definition, which is 1 minus hazard ratio of these hazard ratios. So this one is now a causal vaccine efficacy effect, and of course, it will be good to come up with the good estimators for this, because that is probably of the most interest. Another one, just a variation of that, you may think about that I'm not interested in exactly subjects that I was just defining before, but only in subjects who would not experience the endpoint within the first tau days on both treatments, but will be adherent or compliant on the vaccine. So that doesn't matter what happened on placebo. And then you see that this is a stratum definition, and you can go through the same exercise, define the survival function, hazard ratio, and then definition of the vaccine efficacy. I want to, hopefully I have still time, to go to the vaccine efficacy against severe or disease or death. So like I mentioned, when somebody will be infected, they may have different level of the disease. Some may have mild, some may have moderate, some may have severe, or some may be dead. So if there is then a scientific interest to look at what vaccine is doing in those who will get infected, but what is their, let's say, category? Are they severe, or they die, or they are maybe mild? So this causes a bit of problem, because if you do it naively, looking at just those who have been infected and have this one, that will be definitely a biased estimator. And here I'm trying to set up a setting where you can use principal stratum and find out what will be a causal vaccine effect against severe disease or death in principal stratum. So just you can go easily through this exercise, and then now the vaccine efficacy is defined for this now event, for this variable, is the difference between these 2 proportions. So motivation for PS estimate in this sequence is that we are unsatisfied with a standard not causal estimate that compares the post-infection outcome in the infected vaccine group versus the infected placebo group, because like I mentioned, that would be biased. The principal stratum estimate restricts attention to a subgroup of scientific interest, namely those with no vaccine preventive effect on SARS infection, but then you want to differentiate what the vaccine in comparison with placebo doing on the level of the severity of the disease. Do they have mild, moderate, severe, or death? You can, of course, consider this as a categorical endpoint, but in my presentation, I looked at it as a binary endpoint. Let's say if severe or death, then you are kind of having the severe disease, otherwise you don't have it. So separating these two effects is helpful for designing improved vaccines and for predicting the public health impact of a licensed vaccine. And I think that probably indeed this Estimand answers or is a good answer to a question which has a meaning for regulators, sponsor, but also most importantly for the subjects. So they may ask a question: if I am going to become infected, regardless of treatment assignment, will the vaccine reduce the chance of severe disease death? So I think that that is clearly a good research question, and we need to provide opportunity to evaluate it. So Jürgen, I have some very technical slides here. I believe that I'm already over with my time. I will not go through the developments there, but you can always have a look. And I want to close here with my conclusion. So I try to illustrate that ICH E9R1 provides a useful framework for addressing specific objectives of vaccine efficacy study in systematic and structured manner. Causal vaccine efficacy effects should be clearly defined, and appropriate estimators should be considered, and principal stratum strategy for addressing the specific intercurrent events that I try to highlight in vaccine efficacy trial may be more appropriate than the conventional approaches, either per protocol or ITT. And here are some references, but I want to acknowledge my Janssen COVID-19 vaccine team that I have a chance to work together on this. So I will leave here for the more recent publications on this topic for those who want to go more in depth in details. Thank you very much.
Thank you very much for that, Vlad. That was really, really interesting, particularly recognizing the huge need that all of our societies have for a COVID-19 vaccine. So in the interest of time, we've decided not to take any particular question to Vlad's presentation to give us more time for the panel discussion. So with that, I'm now going to hand over to Anna Berglund, who's going to lead us through that. Over to you, Anna.
Thank you, Jürgen, and thank you everyone for really great presentations. It's been really interesting to listen. So let me start by introduce myself. So my name is Anna Berglund. I am a statistician at AstraZeneca, and I'm also part of the PSI and EFSPI regulatory committee, where I work with Jürgen and others colleagues from across Europe. And I'm very excited about this new joint webinar that we are hoping gonna be a series with regulators and industry from Europe and the US. So this is very exciting. So feel free to send questions, but as we get started with that, I would like to share some reflections from the talks today. I'm really excited to hear that the Estimand framework is being used, and it seems to be very helpful, and for the reason of fostering the proper communications between industry and regulators. I would like to, we already started to touch a little bit on that, and Andreas got a question to that point. But maybe we can start with the industry and my industry colleagues. So what is your reflections? Are you starting to have the proper cross-functional discussions at your companies, or is this considered a statistical issue? So I would love your thoughts.
So again, let me start and then you can complement. So I can share with you experience at Janssen. After release of step 4, we went through a kind of training across functions, not only statisticians, but all the functions which may be affected by ICH E9R1. That was more like to roll out the notion of Estimands and everything around it. We are waiting now to perhaps for M11 when they will develop the new templates of the protocol. So we didn't make an attempt to change our template and to guide the teams where to put, what to put. But of course, in consulting of our statisticians with, let's say, clinical colleagues, we always try already at this point to put perhaps the text about Estimands very high in the protocol section, maybe after objective and where it was previously endpoint. We are recommending them to use that space in the protocol to describe what are the Estimands, how to define the Estimand, and perhaps just a list of intercurrent events, key intercurrent events. And of course, more details then go in the statistical section. So at least this is how we are now running for about 9 months. But yes, like I think John also mentioned at the beginning, that we know that ICH is developing N11, and some of us from E9 R1 expert working group were invited to work together and help them to decide where is appropriate text and how much details should be in the new protocol template.
Yeah, maybe I can add a few points. I mean, certainly we do see clinicians starting talking about this, and some are really excited about that and do see also the opportunities. And I think COVID-19 was actually helpful there for some of them, because there you could really have a discussion on what are the intercurrent events due to COVID-19 on your study. Is your trial interpretation going to be impacted by COVID-19, right? And so on. So I think this was maybe even a good thing to happen if you want to find something positive in that. But certainly it's a long way to go. And I think on both sides probably, there was a similar question, right, to Andreas about receiving questions from clinicians. And certainly on our side as well, if we get an estimate question, it's probably fair to say that at this stage, it would be statistician responding to the regulatory question on estimates. But yeah, things are moving slowly with the protocol, certainly across the industry now, having the estimate wording not in the statistical section, but really in the design objective sections. And yeah, I think we will get there, but it's a long process.
Thank you. So maybe continue with a similar question to our regulatory colleagues. And Andreas, you already answered this, but maybe we could build on that and to see, because my understanding is that you might have the similar struggles as we have at the health authorities. And then also maybe to build on that, are you talking to each other to help us with consistencies across health, regulatory bodies or health authorities?
Perhaps I'll go. So to the first question, I think we are having very productive conversations between stats and clinical teams. I guess Estimands is seen as a topic over which statistics has sort of ownership, but ultimately the questions are clinical, I think. So I would say the role of the statisticians is often to digest the Estimands section and translate it into a topic for discussion with the medical reviewers. And I think we have had many productive discussions in that light. On the sort of cross-regulator collaboration, there's not a lot of explicit collaboration. We have, of course, cluster meetings in different topics, and Estimands may come up at those. Not at ones I've been to, which is a small subset of them. And then there's opportunities for parallel scientific advice, but again, that's not an extremely common mechanism that's used. So I think there probably could be more direct collaboration between the health authorities.
Yeah, I think to the latter, to the collaboration, we don't have much to add. I fully agree that maybe in some cases maybe optimized. So, but in general, I think it's always good to work with examples to really and to explain where the added value is, and really, as John said, try to translate the estimate in the clinical questions. And I think when this is done, I think the clinicians usually, they understand it, and then they have clear opinions. And I think it was really a good experience to work on the regulatory guidelines together with clinicians. But unfortunately, this work is now somewhat on hold. But I think when they understand what it's about, I think we can make some progress. And I think it's, I mean, there are maybe some hurdles, but when we can overcome them, I think there could be some progress. But of course, a lot of, there's also some, reluctance in some areas to adopt the concept, as I mentioned. So there's still some work to do.
Perfect, thank you. Talking about that reluctance, and maybe this, I'm intrigued by Evgeny's talk about, and yours, Andreas, when we talk about, and maybe this is a perception, but that industry is trying to push now and see the estimate framework as an opportunity to maybe step outside of the more traditional ITT framework and start to look at different kind of strategy for intercurrent events. I've gonna clearly specify the hypothetical for overall survival in oncology, and Vlad about the principal stratum in the vaccine trials. And then Andreas, I think you had in your slide that it's also could be seen as an opportunity for regulators to really drive back. And I heard others say this, that to take back what was the intention with the intention to treat, and it's not just subsetting the data sets and then skipping data after intercurrent events and just as you missing at random. So are we coming from different positions, or are we really wanting the same things or talking about the same things, or so I'm curious about that. And then also maybe to Evgeny's point, how can we collaborate and accelerate our journey and our innovation going forward? Anyone can start.
I'll give it a shot. So I think ultimately everyone has the same goal, which is to get safe and effective medications to patients. There are different incentive structures for the businesses and the regulators, which oversimplifying may tend toward, higher probability of approval on the business side versus lower probability of bad approval on the regulator side. But I think the fact that the ultimate goal is the same should lead to common scientific understanding in most cases. I don't think it's not really been my experience that E9R1 is being digested as a way to get ITT further entrenched at FDA. I think it has given us a chance to look at things that we do by reflex and think about which ones genuinely make sense and which ones don't. So ITT is often the right scientific question, or I should say treatment policy, but it's certainly far from always the right scientific question. Having said that, there's, I think, a well-known fear is, I don't want to use the word fear, hesitation to do new things. Whenever people feel that the current system works, there's a strong burden of proof that whatever is new is going to be better. So it's going to be an ongoing growth process, I think.
Let me follow on that. I think that even the fact that this estimate and sensitivity analysis is now an ICH guidance is already a big step forward, because yes, in the expert working group, everybody was represented, and that was a compromise as a product. Now we are in the implementation. I think the key here is what John just mentioned, is a change management. And everything which is new, which is innovative, always will be first reaction resistance, right? I think that through events like this one, and I want to commend the organizing committee, having parties together and explaining where we are at the same kind of in the same place, or where we highlight some potential divergences that Andreas mentioned. Maybe we together can find that solution. And I hope that this guidance or ICH is providing us that opportunity to discuss. I think that at least that is already a big plus, that we have opportunity to discuss, highlight what are the objectives, what are the components of Estimands, and have a useful discussion. That cartoon that John was showing us, when regulators sit and they use Estimands and everybody on the other side also say Estimands, not abracadabra, that is already a plus. I'm very optimistic that we will get there, join the efforts.
Fantastic. Anything to add?
Well, maybe on your second question,
I think...
what we... Oh, go ahead.
No, please.
Yeah, just on the second question, what can we do to accelerate the learning of new things, right? So, John, maybe any thoughts? As you said, yeah, it's difficult kind of to move to something new. So how can the industry also help maybe educate people at FDA or learn together, right, and work with academia as well? What is needed there?
Sure. I don't have anything super specific, but I think that events like these are very helpful. I assume that it's being attended by people from all areas of biostatistics. I think Vlad mentioned case studies. I think someone mentioned case studies. I think case studies are really key. So the primary way we learn is for innovative protocols to be submitted, and then we discuss them, and sometimes the right decisions are made and sometimes the wrong decisions are made, and then there's more discussion, and it sort of iterates in that way. One, I would mention, moving out of Estimands a bit, FDA started several years ago our complex innovative design pilot program, and part of the premise of that was we would review these designs, and also the designs would then be public for public discussion. And I think whatever can be done to sort of bring interesting discussions out into the community is very helpful.
Yeah, what I think is also helpful, if you don't try to go too far too fast, so to say. But I think it's helpful if we just don't try to replace the primary analysis right from the beginning with some new concepts that are very different from what we are used to do. I think it would be helpful to gain experience with some supplementary estimates addressing maybe treatment, the principal stratum, or some hypotheticals, and that we just get more experience and the people get used to it without going too far. So that just getting more experience, so we can, and I think that this will increase the trust in these new approaches.
Can I follow up on that? Maybe, so as you say, primary analysis, so would a study with a primary analysis based on PFS and then overall survival later as a secondary endpoint and be then a good candidate to have their hypothetical estimate, if this is the right setting where really there's a crossover implemented, for example, and it doesn't reflect clinical practice? So it would be the main analysis for overall survival, but it's not the primary analysis for the study, and the approval is granted based on PFS.
Well, I'm not sure whether this would be possible, because I think as regulators tend to be conservative, and I think I would say from the beginning, I would try really to do it as a supplementary analysis for overall survival, and because maybe you are aware that we have I think it's a question on some document, I don't know what the title, what it's called, but it's an EMA document on crossover in oncology, where the estimate framework is also referred to. But at the end, the conclusion is that we do not really trust these methods for primary analysis, because they rely on too many assumptions. And I think this is also something that we should not forget. Of course, often it's easy to agree that we are interested in that specific questions are the one that would be in theory the one of really of clinical and interest, but sometimes it's really difficult and challenging to get a robust estimates of these effects. And I think you will have to find a balance, and nowadays we are not used to make analysis based on so many assumptions. So I think many regulators are reluctant to accept that. So it may change in the future, but for the time being, I think it's still difficult.
So could I build on that, and maybe a little bit on a question that we got earlier? I believe it was or maybe you, Evgeny, was the one who asked that. But there is this communication of patient-centric measures, and John, you alluded to this too. How do we communicate our treatment effect to patients in a good way, and in a clear way, and transparent way, and meaningful way in our labels, compared to what might the data need be and the estimate needed to really evaluate benefit risk, which might be different. Any thoughts on that? We mentioned the non-proportional hazards, but any other reactions to that question?
I agree with what I think Evgeny said, which is that it's not going to be one number that tells everybody everything that they want to know about a drug. That's never been true, and it never will be true. Having said that, I think we do expect there to be one or a small number of core values that are used to communicate. And in particular, in the US regulatory regime, we expect generally alignment between the primary analysis of a primary endpoint or primary estimand and the explanation in what's called section 14 of our product labeling, the clinical studies section, which explains why FDA believes that this is an effective drug. And there should be correspondence between those two things. And so that's sort of where I was coming from in my remarks. If that number is not interpretable to anybody who would be reading it, I'm concerned that it's not the right number to put
there. Good. Nothing to add from anyone else? I don't hear anything. We have gotten some questions around the estimate, the application of the estimate framework outside. I think many of us think about confirmatory trials and the primary estimate and the connection to the label. So what's the thinking about estimates in connection to safety? Evgeny, you mentioned real-world evidence. There are questions around PK studies, biosimilar studies, early development generally. I don't think there's anything saying that we shouldn't use estimate in those frameworks, but is this being implemented, Vlad, Evgeny, do you know from in your companies, or have you seen it a lot in other settings than in confirmatory trials from a regulatory perspective?
I don't have examples from our company, but I think that from the ICH E9R1 expert working group, at the beginning of the text is written, this is the kind of harmonization document focused on the submission and approval. So definitely is focused on the confirmatory clinical trials, but there is a text there that we believe that the framework can be used and applied in other situations. So yes, it was the word efficacy somewhere. We didn't have specifically a clear statement that safety can be, for confirmatory trial, also can be embedded in this framework. But for safety is a bit more complicated because it's not just one endpoint, usually is a complexity of the safety. So I think that if we will be successful in implementing and rolling out this idea for confirmatory studies, others will learn, and if it will find it beneficial, they will apply in other areas. That's my position on this.
Yeah, I can share that within my company, we are rolling it out also to phase 1 and phase 2 studies as well. So people are supposed to be trained as well, and obviously to have discussions on the estimates. And I think it's also important to get the estimate right in early phase study, so that because you take decisions in the end of the day for phase 3 based on phase 1, 2, right? And so you need really to have this consistency in the estimate as well there, and to have a good understanding of the data. And I guess sort of there was a second part of your question, no?
I don't remember.
No. Yeah.
Real world evidence, I think that Anna was asking, you mentioned. So how that
I know, I just wanted to say on safety side, yeah, exactly. I think there were some publications by the SAVI group, I think it's in academia and also industry collaboration on estimates and safety, and I think, yeah, people can take a look, but there is some work definitely done. And yeah, I think there's also common understanding among many of us that just doing proportions for safety is maybe not the best way to describe it, especially in settings, let's say, with competing risks and many other factors.
John or Andreas, anything to add? No? So we have 1 minute to go. So I would like to take this opportunity to thank you all. It's been really amazing. We have a ton of questions that we haven't addressed. So I'm sure there will be follow-up on this. But thank you so much for taking the time. Thank you for being so open in the discussions. And thank you for everyone who has been listening. And I hope you thought it was as good and exciting as I thought. So thank you all.
Thank you, Anna.
Thanks.
Thank you, everybody.
Thank you very much.